introduce our ECHL endowed lectureship today for Grand Rounds. So a person who needs no introduction in this audience, but is the originator of the ECHL endowed lectureship is Dr. Bob Eckel. He's emeritus professor and Charles Betcher Endowed Chair in Atherosclerosis for the divisions of Endocrinology, Metabolism and Diabetes, and Cardiology, Department of Medicine. Bob and Margaret Eckel provided a gift to the Division of Endocrinology, Metabolism and Diabetes and Cardiology in 2022 to establish the Robert Eckel Endowed Lectureship in Cardiometabolic Medicine. This supports an annual lectureship in cardiometabolic health, joining endocrinology and cardiology to help break down silos in medicine and encourage deeper collaborations around connections between diabetes and cardiovascular health. Dr. Eckel has published over 400 articles in this space, and so is a well-known expert, not only locally, regionally, nationally, but internationally. So we thank the Eckels for their generous contribution and sponsorship of this lecture today. It is also my pleasure to introduce Dr. Jose Florez, who is our speaker today. And thank you. And so Dr. Florez is physician-in-chief and chair of the Department of Medicine at Massachusetts General Hospital, Brigham. You can leave it there, sure. He's the Jackson Professor of Clinical Medicine at Harvard Medical School, an institute member of the Broad Institute. Dr. Florez received his BA and MS from Northwestern University, where he remained to obtain his MD and PhD in the medical scientist training program. He then completed his medical residency and a fellowship in diabetes, endocrinology, and metabolism at MGH. Prior to becoming the chair of the department, he was the chief of endocrine division in the diabetes unit. His research focuses on high throughput genomic studies in type 2 diabetes and related traits in multiple international consortia. He is particularly interested in advancing precision medicine and pharmacogenetics as applied to diabetes. He has led the genetic initiatives of the NIH-funded diabetes prevention program, the glycemia reduction approaches in diabetes, a comparative effectiveness study, or known as the GRADE clinical trial, and he designed a study to understand the genetics of the acute response to metformin and glipizide in humans, the SUGAR MGH study. He is an inducted member of the American Society of Clinical Investigation and the Association of American Physicians. He was the 2010 recipient of the Presidential Early Career Award for Scientists and Engineers, the highest honor bestowed by the United States government on science and engineering professionals in early stages of their independent research careers. Additionally, in 2019, he received the Father of the Year Award from the American Diabetes Association. I could go on because Dr. Florez is so well known, but I would like him to be able to get to his talk today. So I'm thrilled to invite him up to the stage to hear his talk today.
Thank you.
Thank you, Dr. Florez.
Thank you. Thank you. So hello, good afternoon, everyone. So it's such a privilege to be here. And thank you so much for the invitation. I'm really honored to be part of this lectureship. I think everybody has scanned their QR code. And I just want to, a word for Dr. Eckel, who I've known for a long time and who has an amazing career that you all know about. We have a personal relationship, so I'm grateful for that. I'm grateful for my name to be joined to his. One thing that people maybe not realize is that of all the human beings that have ever existed, only one in history has ever been the president of both the American Diabetes Association and the American Heart Association. As you can see, hopefully you can see the mouse. And so, it's important for people to understand, all our cardiology friends in the audience, that when you prescribe a beta blocker, that's an endocrine drug. When you prescribe an ACE inhibitor, that's an endocrine drug. And an ARB blocker, that's an endocrine drug. And you want to say everything on a GLP-1s and SGLT2s, those are all endocrine drugs. So statins, of course, are also endocrine drugs. So to have somebody who has really merged the two fields in cardiometabolic disease exemplifies where we are as a field working together for the benefit of our patients. So let's start with just diabetes and the problem we face. Anybody in clinical care who has internal medicine training, whether you're in primary care or any other specialty, you need to know that when somebody comes in with hyperglycemia, you need to make this decision. Is it type 1 or type 2? Hopefully everybody knows that, because type 1 requires insulin for survival and type 2 not always. If you're an endocrinologist, you know that there is a little more complicated than that. There is not just only type 1 or type 2, There is some monogenic forms of diabetes that cause diabetes on the basis of a single base pair change in the DNA. They have some rare forms of insulin resistance. They give you lipodystrophy, or you have mitochondrial diabetes and deafness, or you have maturity onset diabetes of the young. Those are all monogenic. Then you have type 1 classical autoimmune diabetes, but then within that, you have a flavor and polyglandular autoimmune disease where multiple endocrine glands are affected by the autoimmune insult. You have latent autoimmune diabetes in the adult, which is basically type 1 in an older person. You have ketosis-prone diabetes that may present in DKA, but is antibody negative, so sort of an in-between phenotype. And the rest we call type 2. But type 2 is just hyperglycemia that is not monogenic and that is not autoimmune. That's it. But there is a lot of hyperglycemia. And so if you're taking care of patients with type 2 diabetes, you know that in clinic, they come in many different flavors. And that type 2 may be somebody who's lean, and somebody who's obese, and somebody who's old or young, or this ethnic group or that ethnic group. So there's many different subtypes of type 2 subjacent under that hyperglycemia diagnosis that is antibody negative and doesn't have an obvious mutation. But in clinic, we see many, many different types. And we have a very you don't have to read this, but we have a very complicated algorithm to choose how we select a therapy for a patient with type 2 diabetes. And if you think about it, the decisions that get made as to whether you start with metformin or you apply a GLP-1 or an SGLT2 or something else, they're almost never based on the underlying pathophysiology of the patients. They're based on comorbidities. Do they have coronary disease? Do they have kidney disease? They're based on cost. They're based on potential for complications. Can this person tolerate hypoglycemia? They're based on A1C goals, but it's not really about what is the underlying process that led this person to develop hyperglycemia in the first place. So there's a real need to try to be a little more granular. We want to apply precision medicine, and this is true not only for diabetes, but for any other complex phenotype, hypertension. You need to really come up with ways in which we can be a little more granular. And so there's a number of different ways in which we can do that. One way is what you can call sort of an empiric approach. And this has been championed by a group in Exeter in the United Kingdom. Andrew Hattersley leads that group. John Dennis is sort of the up-and-coming investigator who's driving a lot of this. And this is given a database of how people did with different medications. Can I identify what were the initial predictors that led this person to have a better response when I look at, say, for example, glycemic outcomes? So you take 22,000 patients that started sulfonylureas or thiazolidinediones in this clinical practice research database, and then you do your exploration in that initial group of 22,000 people, and then you try to get reproducibility by replicating into some clinical trials and other databases. And so this is a very simple initial experiment where what they said is, okay, sulfonylurea versus TZDs, we're going to look at two axes, gender, male or female, and obesity, BMI, not BMI. And what they're able to see in this group is that if you are, so in the red is TZD response in terms of glycemic response, sulfonylurea is in blue. And if you are a non-obese male, you do much better with sulfonylureas, your glycemic response is much better, your A1C reduction is much better. And if you're an obese female, you do much better on a TZD. And then you can then take to see what happens over time. And you see that over time in clinical trials, non-obese males, again, do initially very well on the sulfonylurea, although over time, the effect of the sulfonylurea is less effective. And then obese females do well on that TZD, and they kind of remain there. So that's empirically, what are the predictors of response based on a clinical trial? And can I be more precise when I prescribe a medication based on what I think is going to happen? So now they've expanded that to the newer generation of drugs. So in the much more recent publication, they've included SGLT2 inhibitors and GLP-1 receptor agonists, and they've come up with an algorithm, which now you can plug into a tool that they have online, where based on whether it's A1C or whether it's in terms of the GFR, the initial GFR, initial A1C, initial GFR, and some of the other characteristics like BMI, is there a distinction whether people do better with the SGLT2s or with the GLP-1 receptors? And again, you can put this as a way of empirically deciding based on these characteristics how I'm going to prescribe. And then finally, more recently, they've concluded this TriMaster study where they did a crossover study with 3 different drugs, pioglitazone, so a TZD, sitagliptin, a DPP-4 inhibitor, and canagliflozin, an SGLT2 inhibitor, and all the groups went through all the different iterations. And the 2 variables they looked at were BMI and GFR. So for BMI and for GFR, they divided this thing, and then how do people do differently? And again, they showed in this TriMaster clinical trial that BMI was a distinguisher in terms of your response to the TZDs, and then for kidney disease, GFR also made a difference in terms of sitagliptin response versus pioglitazone. So anyway, this is one approach in which we use clinical records to apply precision medicine in terms of medication response simply based on some of these characteristics, which tells you still not much about pathophysiology and underlying mechanism, but at least it gives you something that is better than simply using a one-size-fits-all algorithm, which is what we have today. But I think what we need to do is go beyond this empiric approach that is dependent on clinical records and then try to harness the incredible ability we now have with platforms that query all the axes of biology of an individual to see what are all these variables that contribute to the person's biological state. So whether it's your genome and the different variants that you've inherited in your germline genome versus your somatic genome, whether it is how that influences which genes are active in any particular tissue at any particular time, developmental stage, or in a different metabolic state, what is transcribed, what are the messages that get transcribed in those tissues, what are the proteins that are active in cells or circulating and then creating metabolites that you can also measure, and then the microbiome, all these different axes you can query in a single experiment. When you capture the entire universe of circulating metabolites or circulating proteins or DNA variation and combine it with the many environmental exposures that contribute to whether a person is healthy or not healthy. And so there's been various attempts in type 2 diabetes to try to do this in a more patient-centered manner that is about the person's underlying biology. And the one that has generated the most traction so far came out of a Scandinavian group led by Leif Group, which has been working on diabetes for decades. And what they did was something relatively simple. Based on an initial cohort of 9,000 participants in Scandinavia with new onset type 2 diabetes, or new onset diabetes of any kind, they looked at 6 different variables that were easily collected. So GAD antibodies, which you typically use to diagnose autoimmune diabetes, age, BMI, A1C, and then 2 measures, one of beta cell function called HOMA-B, which is C-peptide derived, and then one on insulin resistance called HOMA-IR. And they said, okay, given these 6 variables in this population, can I define some clusters? Do people cluster in a way in which these things tend to define subgroups that I may be able to categorize? And so they came up with 5 different clusters, one which is clearly autoimmune. So this is what we would call type 1 diabetes or LADA, severe autoimmune diabetes. And then 4 that would be kind of logged as type 2. One is a severe form of insulin deficiency, non-autoimmune, severe form of insulin resistance, and then a mild form of obesity-related diabetes and mild age-related diabetes. So those are the 5 clusters. And the way they distinguish themselves can be done by, for example, when you look at things like HOMA-B, which is beta cell function, the 2 that have very little beta cell function are the autoimmune diabetes and severe insulin deficient diabetes, say compared to severe insulin resistance. When you look at insulin resistance, that's the group that comes up, etc. So these groups seem to differ. Now, anybody with any large database can throw variables and come up with clusters. You know, that you can do. Of course, you can cluster data, and there's some pattern, some correlation among the data that may allow you to do that. The question is, does this matter clinically? Does the reflection of these groups lead to maybe different outcomes? And so, for example, when you look at kidney disease, what is really interesting is that people with severe insulin-resistant diabetes in the green curve get chronic kidney disease a lot faster than the other subgroups. So if you happen to be in this subgroup of type 2 diabetes, you're more likely to develop kidney disease. That's in one cohort. When you look at end-stage renal disease, that's the same thing. Completely different cohort. You have the same observation of people with severe insulin resistance progressing to end-stage kidney disease faster. What about medications? So when you apply, for example, how likely are you to progress to insulin? Of course, people with type 1 diabetes start insulin right away. That's understood. But of the other so-called type 2 diabetes, it's clear that the severe insulin deficient people who are antibody negative and would have been defined in clinic as having type 2 diabetes are at much higher risk of progressing to insulin a lot quicker than the other groups. And so now this is the observation of the initial landmark Scandinavian paper. And so the group in Exeter tried to replicate this in a different context. So in 2 clinical trials, in this case, ADOPT, which was a clinical trial where people were randomized to metformin, sulfonylurea, and thiazolidinediones. They look at the 4 different type 2 diabetes subgroups. And it's interesting, for example, that if you have, again, severe insulin deficient diabetes up here on the top left, you respond to sulfonylureas better. That's the red curve, where you do a much better response. But then over time, these people who are losing beta cells and becoming insulin deficient no longer can respond to an insulin secretagogue. On the other hand, if you're an insulin resistant person, you have severe insulin resistance, the best drug for you is a TZD in the blue that stays active through the course of the measurement in the study. They also showed that rather than dividing people in buckets that are categorical, where it's binary, you're in this bucket or you're not, it's a lot better. You get more definition if you apply these predictors as quantitative predictors, as opposed to simply trying to put people in a category. Now, one issue with this, even though it seems to be reproducible in populations and have an impact on clinical outcomes, is that because it's based on measures that change over time, like your A1C or your BMI, the categories are not fixed in time. And so when you see longitudinally what happens, well, this study in Mexico and this study in Germany show that you can start in these plots, you can start in one group, and these curves that you see is how people move from group to group. So it's not like you can put people in a bucket, an initial diagnosis, and say, okay, for the rest of your life, you have this subtype of diabetes, you may change. And so how helpful is it to make that ascertainment initially when 3 months later, you're gonna be in a different group? So that's a limitation of this approach. And so just to summarize this sort of a phenotypic subclustering of diabetes, the strength is that on the one hand, these clusters make physiological sense. They recapitulate what we see in clinic, and they're reproducible across various populations. I haven't shown that, and it seems to have an impact on what outcomes you have in clinical trials. But the challenge is that, again, these phenotypic assignments are contingent on variables that change over time. Some of these variables are not measured in clinic, like C-peptide, not always. And then it's not, one thing is to say I can take a database and divide people in clusters. A very different exercise is to have a patient in front of you in clinic and to say, you individual, you belong in this cluster. To make that assignment with confidence and say you belong in this bucket is a lot more difficult. So what we need is an approach that, again, recapitulates physiology, as this one did, but is not contingent on the time in which the variable is measured. We also need an approach that defines clear disease subtypes that are robust across populations, that can be distinguished from each other, and where I can take an individual and put them in the bucket. And then, of course, they matter. They matter for prevention, they matter for therapeutics, or they matter for complications and surveillance. And what I will posit is that genetics is one way to try to get to that approach, where germline genetics, which is immutable, is there for the life of the individual, can help us do that with a little more solidity. And the example we have to go from is from monogenic diabetes. In monogenic diabetes, knowing the genetics of the person matters deeply, deeply. And the case in point here, I'm gonna mention two. One is neonatal diabetes. Neonatal diabetes is diabetes that occurs in the first 6 months of life. And so, you can imagine the newborn is born, everybody's happy with a new baby, and within 2 to 3 months, that baby is peeing a lot, and the diapers are heavy, and they're irritable, and they're not consolable, and now you go to clinic, and their glucose is 300. So that family now has to cope with a diagnosis of diabetes where the therapy available to that baby was insulin, and so you try to do insulin in a newborn and see what that's like, or an infant, or then a child, an adolescent, multiple glucose measurements a day, multiple daily injections. Not an easy task. But it turns out that neonatal diabetes is not caused by autoimmunity, it's caused by mutations in the sulfonylurea receptor or the potassium channel that transduce the glucose signal to insulin secretion. So for those of you who are not familiar with how this works, right now I think everybody's having a sandwich, and the sandwich has carbohydrates, and that is being metabolized into glucose, and that is being actively transported into your beta cells by this glucose transporter. And upon entry in the beta cell, it's phosphorylated by glucokinase, which serves as the glucostat of the organism. And so the phosphorylation of glucokinase from glucose to glucose 6-phosphate makes glucose 6-phosphate into the Krebs cycle, you make ATP, and then ATP binds the cytosolic side of this potassium channel, which at rest in the fasting state is open, allowing potassium to go across the membrane. When you close it, you retain potassium intracellularly, and that depolarizes the membrane and activates voltage-sensitive calcium channels with calcium entry and very quick calcium-mediated exocytosis of insulin molecules. Mutations in the potassium channel sulfonylurea receptor prevent this from happening. So the person has diabetes because there's no way for them to make insulin in response to glucose. And the observation that this could not be autoimmune came from a clinician, Dr. Hattersley, who was also a geneticist. And so, true story, the way this happened is a man in Brazil, born in rural Brazil, in a resource-poor setting, was diagnosed with neonatal diabetes in the first 6 months of life, and put on insulin as you would, by the assumption that this is autoimmune destruction of beta cells. So this person took insulin for the first part of his life, and then eventually got tired of insulin. And then when he was in school, he chucked away all the syringes and no more insulin for me, and I'm tired of this whole thing. And something amazing happened. the person survived, did not die, which you would if you're not taking insulin and truly have autoimmune destruction of beta cells. So then went on to law school, became a man of means, gets married, has 2 children. Now the 2 children have neonatal diabetes. So then being now a very smart person says, well, I have it, they have it. I think it must be genetics. So then now that I have means, I'm going to find the world's expert who's going to tell me what is happening in my family. And so he flies to England and meets with Andrew Hattersley. And Andrew Hattersley says, well, you can't have type 1 diabetes if you survive without insulin for several years. And the mutation had just been found. And then the observation then is, it's not that your beta cells are destroyed. We still have all the machinery here. The only thing that happens is that this molecule needs to be closed, and we can't seem to close it because of the genetic mutation. Well, maybe if we take the medication that is supposed to close it, as sulfonylurea, but give you very, very high doses, we might be able to overcome the genetic defect and for the first time in your life, close this channel. And so a study gets designed for this person, and there is a New England Journal paper in 2004. How many people were alive in 2004? I have to be very careful when I ask that question, because increasingly there are some who will not raise their hand, but hopefully here, most people are alive in 2004. And N of 3 for this New England Journal paper. You can publish a New England Journal paper with 3 people, proof of concept, where they're given glucose and they don't make any insulin because they never have for their whole lives. But then they're given tolbutamide, which is an insulin secretagogue, and IV sulfonylurea, and they have this robust response. All of a sudden, their beta cells wake up and with a single injection are able to make insulin. And so then a clinical trial follows where you take a number of people with these mutations with different A1Cs at baseline. No one wanted to be randomized. So this cannot be a randomized trial because no one wanted to be randomized. Everybody wanted the drug. And all of them in this observational study responded very nicely with their A1C coming down to baseline, showing that you can improve not only their quality of care because their A1C is better, but the quality of life because they no longer have to take insulin injections or depend on daily glucose measurements. They can take a single pill once a day and have their diabetes addressed. So knowing the genetics, this is precision medicine applied for this boutique, rare form of diabetes. And if you want to pick me up, if you're an endocrinologist and you want to pick me up, just go to YouTube and look at Journey to a Miracle, where you see how this happened for Lily, who's a patient at the University of Chicago who underwent this transition. So that's one example where precision medicine happens. The other one in monogenic diabetes and MODY diabetes, which is about 2% of all diabetes, is when you have glucokinase mutations. So I mentioned that glucokinase is the glucostat of the organism. Its activity is dependent on the ambient glucose concentration. And loss of function mutations in glucokinase will say that the enzyme only works because it's haploid sufficiency. You can have the other enzyme is fine. It only works at higher glucose concentrations. But otherwise, the entire machinery of the beta cell is intact as well. It simply starts the cascade of insulin secretion instead of at a glucose of 120, maybe at a glucose of 150. So these people will live at slightly higher hyperglycemia, but otherwise it's not a form of diabetes that poses any problem. So when you look at the cohort, for example, over time, and you have people with glucokinase mutations in the dark dots, blue dots, and then their first degree relatives without the mutations in the orange, and you look at A1C by age, you see 3 things. Number one, of course, people without the mutation live at a lower glucose than people with the mutation. So in terms of A1C, you have A1Cs ranging from 6.5 to 7.5 if you have the mutation and lower. So that's one observation. The second observation is that over time, people get a little more hyperglycemic, whether you have the mutation or not. So the slope is uptrending in both cases. But the most important observation is that the curves are parallel. They're parallel. These are not people whose diabetes is going to escalate, who's going to get really out of control, who needs more and more therapy, and who develop complications. And when you take people with those mutations and you ask them what do they have as far as complications, the only thing they have is non-intervenable, non-clinically significant, mild background diabetic retinopathy. They have nothing else. And so unless they are pregnant, which poses a different issue, you don't need to treat them because they have no other complication and therefore no need for therapy. 2% of people with diabetes have glucokinase diabetes, probably undiagnosed in most cases. Many of them thought to have type 1 on insulin. Some of them thought to have type 2 on metformin, both unnecessary, both unnecessary. So it's important to find out on the monogenic side, whether genetics can drive therapy. So what I'm interested in, what we're interested in our group is to see whether the plethora of loci associated with type 2 diabetes, the complex polygenic form of type 2 diabetes, which in the last decade has yielded hundreds of loci across the genome, where a genetic variant increases risk of diabetes, together explaining more than 50% of the heritable component of the disease, whether that matters for prediction or for clinical decision-making. So one of the initial questions we had, and this was work led by my colleague at Mass General, James Miggs, was to say, well, this genetic information that is surfacing, is it better than clinical predictors in determining whether somebody is going to get diabetes or not, type 2 diabetes or not? And so the way you ask that, of course, is you have a clinical prediction tool, and then you superimpose the genetics on that, and you see how this receiver-operator characteristic curve does. A test, a clinical test that is useless when you plot the false positive rate with a false negative rate, and a useless test, a coin toss, head or tails, is the diagonal. It's 50% area under the curve, so you want a curve that approaches 100%. So in this case, a clinical predictor in the Framingham Heart Study that includes common variables like age, sex, family history, glucose, body mass index, triglycerides, and HDL gives you 90% area under the curve. That means that if you have 10 patients in front of you, 9 out of 10 times, just based on clinical predictors alone, you can guess who's going to get diabetes in the next 8 years. And so on top of that, you superimpose the genetics that we knew at the time, a polygenic score at that time of 18 variants, and you see that the curves are superimposable. You get really no improvement in clinical prediction with the genetics, and that is because glucose is a great integrator. You know, when glucose is in the prediction model, of course it's going to predict diabetes. It's sort of like our friend Leif Group would say, it's like predicting rain when you feel water. So it's sort of kind of all the same thing. But this motivated a New England Journal paper for James. It was his first New England Journal paper. He was very, very happy. And this is a true story. 2 months later, I go to Spain to visit my family, and I'm in Madrid. There's, for those of you who've been there, there's a Prado Museum in Madrid. All the Renaissance and Baroque painters have their collections there. And Velázquez is my favorite painter, realistic painter. And then there's this thing called the Drunkards. And it's amazing the resemblance between James and the main person getting drunk. You know, this is sort of how it happened when he got his publication. It's like totally, but it's amazing. It's like this is not Photoshop. You know, there's some model that served Velázquez in 1600 to sit here and was probably some distant ancestor of James. So then we can, then we can construct bigger polygenic scores. So I told you one way is that you take all your results and you construct maybe a polygenic score that takes only the variants that reach genome-wide significance. And I've told you that's not particularly helpful. You can try to go deeper in the distribution and take a lot of variants that are deeper there. And when you can compare how good are the two methods, they're actually also not distinguishable. When you only take the genome-wide significant or a much larger swath of variants under the genome-wide significance threshold, you don't really do much better. And that is because the ROC curve for genetics, again, it's only mild for type 2. The one exception is in type 1. So in type 1, because a lot of type 1 diabetes is driven by the HLA region, which contains 50% of the genetic prediction for type 1, the ROC curve is much better. And so in some confusing clinical scenarios, for example, an older person who's lean and therefore seems like maybe have autoimmune diabetes, but is antibody negative, so I don't know whether it's type 1 or type 2. Or a younger person, so by age type 1, but then obese, so maybe type 2, but then antibody positive, so maybe it's a false positive. You can actually do a polygenic score for type 1, and it can discriminate. And so when you plot the type 1 score, the type 1 people and their scores, people with monogenic diabetes or people with type 2 or no diabetes at all, and you set a threshold here, people who score above that threshold are very unlikely to have monogenic diabetes or type 2, maybe more type 1, and people who score below this threshold are less likely to have type 1 and you can have the others. So the use of a type 1 diabetes score, which I know is something that is available to you here in Colorado because of your biobank, and it's a matter of a clinical study that is now being led by 2 of your investigators, will help, I think, determine to what extent this polygenic score for type 1 diabetes can be useful in the clinic, because again, the ROC is much better. What happens in type 2, and this was exemplified by Mark McCarthy, one of the leaders in our field, in this palette model. To simplify, if you have a number of risk factors, each one of a different color, and then you see how they go into this palette, the painter's palette, there may be some people where most of the diabetes is driven by one risk factor in the red, but the majority are going to be in the middle of the palette, which is a composite of many different colors. And it's very difficult to say in the middle, in the center of the palette, whether this is green or brown or gray or what color this is, because it's a mix. So for precision medicine to work, you need to identify who are the people who sit on the edges of the palette, where most of their diabetes is driven by things you can quantify, understanding and acknowledging that there may be a bulk of people for whom that may not be possible. One exception is special populations. So the Inuit in Canada and Alaska and Greenland have lived under extreme climatological conditions for many, many generations. And it turns out that they may be enriched for variants that have led to have risen to prominence. And so they have one variant in the TBC1D4 gene, which is a stop codon that raises diabetes risk tenfold and glucose values twofold, and it has a 17% frequency. And it turns out that it interferes with the ability of insulin signaling in muscle to lead to glucose absorption by the GLUT4 transporter, conferring insulin resistance. So there may be some instances in which special populations may be at the edge of the palate, and in these people, 1 in 5 is going to have diabetes on the basis of this genetic mutation, and maybe an insulin sensitizer is the best drug for them. So a different way to construct polygenic scores then, and we'll get to the crux of the talk, is to then, I've told you about constructing a polygenic score based on genome significant variants or maybe a graded distribution, but a different way is to try to use physiology to drive the construction of process-specific polygenic scores. And this is work led by Miriam Adler in Boston, who trained with me and is now an associate professor, and has led to the definition of clusters that are anchored in genetics but informed by physiology. And so when you apply this, what you do, the exercise is as follows. You first take all the type 2 diabetes genetic variants that different GWAS studies and sequencing studies have discovered. So now you have a catalog of maybe several hundred variants. And then you have a number of traits of interest that have something to do with diabetes, whether it's BMI or fasting glucose or insulin levels or lipids. And you collect all those variants and all those traits, and you know the effect of those variants on those traits from GWAS for those particular traits. So then you do a cluster analysis in database and you come up with potential pathways. So you in effect have a matrix where you have all the risk variants as your rows, you have all your traits as your columns, you run your non-Bayesian matrix factorization algorithm on this, and you come up with two submatrices where one axis is the number of variants by the X number of clusters, and then the other one is the X number of clusters or K number of clusters and the traits. And in this heat map, the intensity of each one of the blocks tells you to what extent that variant or that trait contributes to the construction of the cluster. So when you do this in the first iteration, you come up with 5 clusters, which I will describe next. So you have 2 beta cell clusters, which are defined by low levels of fasting insulin. So beta cells are down, they're not making enough insulin. The genes that belong to each one are genes that have already been implicated in beta cell function. And so one cluster is an insulin processing and secretion cluster. The other one is an insulin synthesis cluster. And then the other 3 are all insulin resistant, where the dominating trait is higher levels of fasting insulin. So insulin is high because the tissues are insulin resistant. The first one is clearly the traditional form of obesity-mediated diabetes, insulin resistance with genes that are, have a role in obesity. The other one is not about fat accumulation, but about fat maldistribution. That's what lipodystrophy is. Lipodystrophy in the monogenic form is about fat not going to adipocytes, which are equipped to deal with fat storage, but going to liver or going to muscle and causing insulin resistance there. And so they have lower BMI, but then fat in the wrong places, and then a liver lipid cluster where you have a liver accumulation of lipids. So that's fine. We've taken genetic variants, we've taken traits, we've gone in silico and been able to come up with these clustering associations. Does this have an impact on biology? And so in a completely independent data set, which has to do with what parts of chromatin are open in what human tissues, then we can ask, given that we have these genetic clusters, do those genetic variants, are they enriched in the parts of the genome that are active in the tissue of interest? And so you have the 5 different clusters and you say, what tissues are enriched for open chromatin along the variants that define this cluster? You see that beta cells and islets are the ones that are significantly enriched. And when you go, for example, for obesity is pre-adipocytes. When you go to lipodystrophy is adipocytes that are enriched. And when you go to the liver lipid cluster is the liver. So a completely independent exploration and a different data set of human tissues that tell you that this biology seems to matter for these forms of diabetes. So then the next question is, does this have a relevance on distinguishing humans in populations? So we take 4 different populations of different kinds. We then have everybody, you can define a genetic cluster for each human being because all the human beings, you know what the variants they carry. And then you can say, well, for the people in the beta cell cluster, let me just take the top 10% for each one of the clusters that are basically the extremes of the palette, the ones in which the color is very well defined. And then I'll calculate the person's polygenic score and then compare them and see what their traits are like. And you see that, for example, people who are at the end of the beta cell cluster, they have much lower C-peptide numbers shown here in the yellow. People in the obesity cluster have higher BMI than their counterparts. People in lipodystrophy have a lower BMI, and people on the triglycerides have lower triglycerides as expected. So you can make distinctions. And in fact, you can ask how many people belong on these extremes. About, how many people are on the edges of the palette. About 30% of all participants have belonged to at least the extreme of one cluster. Well, does this matter for clinical outcomes? So then you go to something like the UK Biobank when you have longitudinal follow-up on 500,000 people, and then you can divide again genetically where people are in their clusters, and you see that when it comes for hypertension, systolic and diastolic blood pressure is a lot worse in people with obesity and lipodystrophy clusters. So again, their complications matter depending on the type of diabetes you have. When you look at things like GFR, it's lower in the liver lipid cluster by a difference with the others, and then coronary disease is also different. So it seems that hard clinical outcomes matter and change depending on what cluster you belong to. Another piece of this approach is that allows you to solve some of the epidemiological observations that have escaped explanation for decades. So now we have 12 different clusters in a multi-ethnic context. So we've expanded the number of clusters. And then one observation is focusing on these two, the lipodystrophy and liver insulin resistance, is can we explain why people of East Asian descent develop diabetes at a much lower BMI than people of European descent? That's a clinical observation where that happens. And when you look at these two clusters, it looks like in 5 different continental ancestry populations. In blue are people of East Asian descent, so Chinese, Korean, Japanese, and they are enriched for people for these two clusters. That seems to be a lot more prevalent in those populations. And so when you plot diabetes prevalence for BMI and Europeans in the red, for a BMI of 30, you have 9% diabetes prevalence. And you say, at what level does it happen for East Asians? For a BMI of 24, you see the same prevalence of 9% diabetes for a BMI of 24. And then you ask, well, once I take into account this genetic makeup that these polygenic clusters are defining in this particular population, then I can explain 63% of that difference, so 2/3 of that difference. So when you adjust for that, then you see that the East Asian curve approaches the European curve, saying that the reason that people of East Asian descent are more likely to get diabetes at lower BMI is because the variants that drive diabetes in that population, for the most part, cause diabetes in this fashion. So this is a method in which genetics, informed by physiology, can lead to clusters that have a difference in clinical outcomes and that can separate populations. For completeness sake, I will just talk about 2 other approaches that other groups have pioneered. This archetypes approach, very quickly, I will just explain. It was about 700 people in this very deeply phenotyped cohort in Europe. They looked at 32 clinical variables, many of whom are not routinely available in the clinical record. The bottom line is that these clouds that you generate, 472 of the 700 people could not be placed with confidence in any cluster, meaning that this is the center of the palette. These are all the people who cannot be placed, and so not super useful, but maybe on the edges. A more appealing approach is something called this data dimensionality reduction, where you construct a tree. So there's a group in Scotland, and what they did is with 23,000 Scottish participants, they looked at 9 clinical traits that are easily obtainable in the clinical record, so HDL and LDL cholesterol, systolic blood pressure, transaminases, etc. And then they can create a 2-dimensional tree where in the branches you have people who have extreme features of any of the 9 traits they measure. The trunk is the shared features. And then the advantage here, you're not putting people in buckets. You belong to this, yes or no. You have a continuum. It's a tree, it's a shade, it's a gradient. And so what you can see in this tree, that the upper right of the tree are people with higher blood pressure and higher total cholesterol with moderate A1C. And in terms of complications, they're more likely to get diabetic retinopathy. And in the lower right of the tree, you have people who have more BMI and triglycerides, more metabolic syndrome, and they're more likely to get a composite cardiovascular outcome or CKD and glycemic deterioration. So this is an exercise in Scotland. And then a group in Germany has done the same thing. And of course, the scientific method is about reproducing data that other people generate and making sure that it's robust and it stands in different environments. And now in a cohort, and these German studies are very deeply phenotyped, they apply the same thing. They get a tree of this kind of the same shape, but they have very refined measures of insulin sensitivity and insulin secretion. And so what they see, for example, is that people who have very low insulin sensitivity are in the bottom part of the tree right here, and people with low insulin secretion are in the top part of the tree. And now they can ask what happens to them based on their deficiencies. And so if you happen to be in the group that is very insulin insensitive, you are much more likely to have a bad cardiovascular outcome, or to have all-cause mortality, or to have diabetic foot disease. So this is peripheral arterial disease. So large vessel disease is much more likely to happen in the insulin resistance people defined in this manner. and then you have a beta cell deficit, you're much more likely to have peripheral neuropathy, diabetic peripheral neuropathy, autonomic neuropathy, and requirement of insulin, as you would predict. And then kidney disease is kind of mixed between the two. So again, showing that this way of defining people based on phenotypic characteristics is reproducible and has an impact on clinical outcomes. So trying to expand this approach to all of diabetes, I just want to mention the RADIANT studies, a study in which this site is one of the sites led by Neda Razuli here. And what we're trying to do in this NIH-funded consortium across the country is to identify 800 people who have atypical diabetes. They're undergoing whole genome sequencing. They're being deeply phenotyped. And there's a number of different study groups that are looking at what happens to them. So one initial finding that was reported recently is people who are antibody negative, but have a hypologenic score. It seems like there is an emphasis on beta cell function and insulin use. And so this is a way in which we are trying to use atypical forms of diabetes to really define the disease in greater depth. So just to conclude, there's a model in which predictive prediction, I think, is going to work. I think genetics provides a skeleton. Genetics is relatively immutable, particularly germline genetics. You inherit it at conception, and you have that for the rest of your existence. And so that's solid. It doesn't change, and you can serve by the skeleton. But of course, somebody is not only defined by the skeleton. You need other things, because with the same genetics, metabolic states change through life. And so I can envision muscle maybe is environmental modifiers, how much exercise, what is your diet, that need to be superimposed on the genetics to be able to predict. You can have circulating biomarkers that give you like C-peptide that give you a sense, or adiponectin that gives you a sense of what the metabolic state is in the person. And you can, of course, in humans, ignore behavior that has a big impact on what happens. So all of these things need to be captured. All of these things need to be placed in prediction models that eventually give you a holistic way in which you're going to be able to apply precision medicine. And so the future that one may imagine is that everybody, and at least in the developed world, will have a genome. Obtaining a whole genome, not just that GWAS, not just a chip, but sequence the entire genome that took decades for the Human Genome Project, can now be done very quickly for less than it costs to get an MRI. So imagine all the MRIs that you all get here for things for a headache. Well, you can get a genome once in the life of the person, and now you have that information. That'll be stored in the medical record. And so then the question will be, what part of that information is actionable? And some of what we're doing, some of us are doing in our field, is to try to define the instances in which having that genomic information actually makes a difference in clinical care, because it changes outcomes, or it's cost effective, and it can actually improve prediction, and hopefully do that in an equitable manner that doesn't magnify health disparities. So the person, will be born, the person will have a genome, and then that will be deposited in the healthcare system that is taking care of that person. In this case, my healthcare system is Mass General Brigham. And then that is all stored in a particular chip. Or somebody may come, from the world with a genome from a different system, or through, entertainment genetics. You've done your genome because you wanted about your ancestry. And be very careful when you do that, especially in terms of family relationships. You find you have relatives you didn't know you had. And so all of that will come. And then what we need to do is then at the point of care, after we generate the evidence that says this is important for diabetes or for hypertension, for cancer, these are the things that matter. And then it'll be stored in the clinical record compared with all the other things that get obtained, so that then the clinician at the point of care, somebody comes in with hyperglycemia, then that person can simply say, okay, here's the algorithm. You have high blood sugar. This is the type of diabetes you have. And this is the therapeutic decision that I will make based on your type of diabetes in terms of surveillance for complications, or in terms of the medication that I would initiate. And then you share that, and you can share that with the clinician, or for prevention, you can share that with the clinician. And so the idea would be that the human genome, which was not available when most of us were born, and is available now at our fingertips, is a treasure trove for humanity. It belongs to all of humanity, and our ability to query it, and our ability to ask whether it makes a difference in medicine, I think will define the way medicine is practiced in the coming century. So the take-home messages from today's Grand Rounds is that type 2 diabetes, hopefully you've learned, is a heterogeneous entity. It's not just one disease. That current treatment approaches that are based on empiric algorithms almost never consider the underlying pathophysiology of the person, and never look at the patient in front of me and say, what kind of diabetes do you have? I look at their comorbidities and other factors to help me make my decision. I think I've shown that whether you use phenotypes, whether you use electronic medical record information or genetics, you can parse out type 2 diabetes subtypes. And these subtypes matter. They differ in terms of outcomes, and they differ in terms of how people respond to therapeutic interventions. Now, whether you should do categories and say you have flavor type 2A or 2B or 2C, or simply use these predictors as quantitative predictors for an outcome as as some sort of quantitative gradient, I think that remains to be determined. But in general, a continuous scale seems to be more powerful than simply trying to place people in categories because everybody's a continuum. I think genetics can serve to anchor subtypes, but it's not sufficient. I think they need to be informed by physiology because the subtypes may not be stable over time. And finally, I think one thing that I didn't spend much time is that the subtypes actually maybe helps open new venues for drug companies and biotech to explore for therapeutic intervention. So with that, I just want to conclude. I want to make it clear that a lot of the work that I presented was done by Miriam Adler and others in the group, and all the clustering work. It's significant that all of this was in silico. In other words, pandemic, yes or no, you can sit in front of a computer and derive insights that really help your understanding of disease. And a lot of that is made possible because all the GWAS data, not only for diabetes, but for all the other traits that matter in metabolic disease, were generously deposited by investigators in public databases that are accessible to the public. So a lot is about public sharing of data that enables this type of work to take place. We have a large group in Boston that contribute to this, a lot of mentors, a lot of consortia and collaborators across many different international groups, and then many funding agencies, and then our own project at home with 4 great daughters. We specialize, have one approach to our genetics in our family, and physician scientist wife who's an ophthalmologist, who's also bilingual in Spanish. And for a diabetologist, it's so great to have a Spanish-speaking ophthalmologist next door to whom I can send patients who only speak our language. So thank you very much, and I'm happy to entertain questions.
Thank you so much, Dr. Florez. That was fantastic. We have some time for questions, so I'll open up the floor to any questions you have for Dr. Florez. Yep. Dr. Eckel.
Hey, Jose, so well done. Thank you so much. The question I have, I think, is probably not easily answerable, but when do you think epigenomic signatures may replace genomic signatures in terms of prediction of disease?
Well, I'm not sure that they will replace. I think epigenomics gives you much more information because it tells you what part of the genome is active, in what cell type, at what metabolic state, in what developmental stage. So it has a lot of information, and it tells you of the genetic variation that you see, when does it appear? Because, for example, if the genetic variation that we identify in a GWAS is only active in the first 3 months after fertilization, when the pancreas is being developed, that is really not actionable in terms of preventing that person's risk. So the epigenomics gives you a sense of where and when to be able to intervene, but it's much more difficult to obtain. And so, obviously, DNA variation is easily accessible from peripheral blood, and you can do that once, and then you have that information. And that's why the GWAS have exploded. Trying to do genome-wide epigenomic studies in the particular cell type, and the particular metabolic state, in the particular developmental stage is challenging. Maybe you can get adipocytes, you might be able to get hepatocytes, try to get beta cells, try to get neurons. So very difficult in humans. We have a project going on that the NIH is funding, a consortium where we're taking 4 cell types, so beta cells, adipocytes, hepatocytes, and myocytes, and then try to build the relationship between genetic variation, inherited genetic variation, and epigenomic states in cells. But of course, that's only the beginning of the process.
A second question, in all the different phenotypes and genotypes that relate to type 2 diabetes, is everyone demonstrating a defect in glucose-stimulated insulin secretion? What's the difference between obesity and type 2 diabetes? It's the beta cell, is it not?
Yes, I think they do, but to a different degree, and maybe not as the inciting event. So maybe at some point they get diabetes because their beta cells are not able to make enough insulin to be able to maintain glucose in the normal stage. But whether that's a reaction to an initial insulin-resistant insult in one subgroup versus another group, which is a primary beta cell defect, I think that remains to be seen.
Do you think that suggests that the beta cells should be a more central focus of investigation, or do you not believe?
Well, one thing, the only way you're going to preserve glycemia is if we reverse the degeneration of beta cells. We have no medications that cure diabetes. We don't cure diabetes. You are a diabetes doctor, you got a patient, it's your patient for life, unless they get bariatric surgery and they go into remission, which is very rare. So for the most part, we don't have disease-modifying therapy. So salvaging, preserving beta cell function is essential to maintain euglycemia. And that is a big active way of investigation. But I don't think that's the only thing that contributes to complications. So I think it's fine to maintain glycemia, but I think the insulin resistance phenotype, even if you have very good beta cells, is still contributing to coronary disease and peripheral vascular disease and putting you at risk for kidney and stroke and everything else. So I think it's more than one disease process. It's, there's a combination and you need to address all of them. Neda.
Yeah, a great talk, thank you. My question is regarding the data you showed on clustering type 2 diabetes, either based on genotypic or phenotypic information. You showed some data on the course of the disease, complication, they might be different, but do we have any data that this clustering will affect our treatment or will be meaningful clinically in our practice?
So it was mentioned in the introduction that we are doing the genetics work in the GRADE clinical trial. So GRADE is a NIH-funded comparative effectiveness clinical trial in the United States where 5,000 people with type 2 diabetes, stable on metformin, were randomized to 4 different drugs on top of metformin. So glargine, sitagliptin, and glimepiride, and liraglutide. So 4 different classes. And then follow for metabolic outcomes and for complications. And the 2 papers were published in the New England Journal about 2 years ago, showing that glargine and liraglutide were better for metabolic outcomes and probably a signal for cardiovascular as well. So we are doing the genetics on that cohort, and we are looking at whether these clusters that we've had defined genetically affect how people respond. We haven't seen a very screaming signal on the clusters. We do have individual findings for individual parts of the genome that seem to affect response to one drug versus another, but we haven't really seen that the clusters are definitive in that setting. So, that sort of approach needs to be expanded in all the cohorts that are available to us to do pharmacogenetics, and then we need to see whether they matter or not. It could very well be that they don't matter. And then that's fine. And then we go with the empiric approach, and we say things like obesity and gender or GFR or baseline A1C are more important predictors of response. And maybe that's what we do instead of the genetics, and that's fine. But I think it's sort of like when you're at JFK in the 1960s and the moon is right there and you think you have the technology, well, let's see whether we can do it, and maybe we get there, maybe we don't, but we need to try, and hopefully we get there. So the same thing with the genome. We have the genome, and let's ask the question, can we actually harness the genome to answer how we're supposed to treat people? And maybe we will in some cases, and maybe in other cases it won't pan out.
So I have another related question to your just recent answer, kind of because this is a general medicine audience. Before we have access to all the genetic information, is there something that you would recommend from a clinical standpoint for as you're seeing these people in clinic? So, GRADE is a great example of you start with metformin, but what's the best next one? And as you so eloquently showed us, not everybody's going to be the same. So sometimes with newer drugs, and especially ones that have really great outcomes, we lean towards one class. But do you have an approach where you kind of give people a trial for a certain period of time to see if they are somebody who's going to actually respond to this class relatively well, and if they don't, then you switch? What's your practical approach?
So I would, no matter the field that you're in, all these decisions that we make clinically are based on averages, based on some clinical trial, and so on average, this population responded better to this drug than to the standard of care, and so we use the drug. It's an average, and around that average, there's a heterogeneous response. So I would say in clinical trials where that is wide, that tells you there's a lot of heterogeneity. And my wife does uveitis, so it's inflammation in the eye, and uses all these biologics and all this stuff that rheumatologists use all the time for autoimmune disease. And it's trial and error. Okay, let's try this one, and then we infliximab and teplizumab, and you'll do all the different things, and it kind of all depends as to what's going to work, and it's more or less empiric. I would say in clinical trials where you have a lot of divergence and heterogeneity, it's important to then start drilling in and saying, let's find a better way of defining what subgroups are underlying these responses, so that there are some people who are not going to respond to the drug, even if on average it was shown to be a significant effect. So look at your phenotype, and especially when the pathophysiology is not very well known, and I can think many areas of medicine where that's the case, then look at the phase 3s and see in what phase 3s there's a lot of heterogeneity.
Great, thank you. All right, with that, I want to really thank you
Thank
for
you.
such a fantastic, well, both endocrine grand rounds and medicine grand rounds today. We look forward to the future with integration of genomics into diabetes care, and